All right. Hello, everyone. Good morning, good afternoon, good evening. Welcome to Advancing Malaria Drug Discovery webinar, sponsored by Collaborative Drug Discovery. My name is Gang Liu, a Senior Program Officer on the Drug Discovery team at Gate Foundation. It's my great pleasure to moderate this webinar. We have a fantastic lineup of speakers for the conversation. We have Jeremy Burrows, Brice Campo, James Duffy, Dominic Bosan from Medicine for Malaria Venture, Professor Kailash Bali from Holistic Drug Discovery Center in University of Cape Town, Professor Elizabeth Wenzler from University of California, San Diego. Last but not least, Mariana Vascati from CDD. And we have a packed agenda and a comprehensive list of topics to share with you. So without further ado, let's start first with Jeremy. Jeremy, so you've been with MMA for almost 25 years. Maybe you can share with the audience on some of the key statistics on malaria and introducing MMA, since many of the folks on this online are not familiar with both subjects.
Okay, thank you, Gang. And in fact, I've been with MMV for just over 17 years. MMV's been around for 25. So the first thing to say is that malaria is still a problem. It's still a very pressing challenge. And these are data from the World Health Organization report from the end of 2023. There are still over 250 million cases and over 600,000 deaths. And two things to really stress is that the burden is in Africa, and there are particular vulnerable populations that we're really focused on. That's kids under the age of 5 and pregnant women. So as Gang said, I work for Medicines for Malaria Venture, MMV, and 3 of my colleagues are on this call as well. So MMV is a Swiss not-for-profit foundation with the mission to discover, develop, and deliver new antimalarials for the world's poor. Now, importantly, we do not have any labs, so everything that we do is in collaboration. And so it's extremely fitting that we're having this webinar with 4 of our important collaborators. And so the Gates Foundation, UCSD, H3D, and CDD Vault.
Thank you, Jeremy. There's some sombering statistics on this slide, right? And in last century, we thought we were close to eliminating malaria, but once we took the pedal off the pedal, parasite roars back. And what can you tell the audience in terms of the discovery and development pipelines that MMV is working on?
Okay, yeah.
And some of the challenges.
Well, yeah. So we still face, from a drug discovery and development perspective, a range of challenges. And these start with the issue of resistance. And this is principally to the artemisinin combination therapies, the ACTs. And I guess especially to artemisinin itself, and also lumefantrine, which is the partner drug to Artemether in the mainstay therapy Coartem. So this is a big focus of what we do. We are really pleased that there are now 2 vaccines in the prevention of malaria and actually provide additivity with drugs. But there are still new and better interventions that are needed. we're really desperate to improve options for pregnancy and also to find non-acetylcholinesterase anti-relapse agents that don't cause hemolysis in Vivax patients with low active G6PD. And then I guess the last challenge really is that we're passionate about the idea of elimination eradication. And to do that, we need to break the life cycle. And so we need to block transmission to and from the human.
Thank you, Jeremy. Yep. Sorry.
If you can move to the next slide, just to really how we're tackling resistance, to follow on from your other question, Gang. In essence, we have a three-pronged strategy, which is over time. And the first focus is really on how do we address the problem of artemisinin resistance. And we've got a range of approaches to do that. One is this idea of multiple first-line therapies, effectively different drugs, which have different partner drugs within the combinations and can tackle resistance that way. We're also actually moving from combinations of 2 molecules to actually 3, to triple combinations. And that's a further strategy. And we've got a very nice project with Moro, Bosan, Marabini in phase 3. Adding single low-dose primaquine is an important component because primaquine kills gametocytes, which actually transmit the parasite. So if we add in primaquine, we can actually kill the resistant gametocytes before they actually transmit. And importantly, and others will talk about this, we have a very exciting non-artemisinin combination therapy in phase 3 with Novartis, which is ganaplacide lumefantrine. In this, the second part of our strategy in the medium term is to really focus on lumefantrine resistance. Again, artemethyl lumefantrine make up Coartem, which is really 75% of the market in Africa. And the lumefantrine resistance strategy really is to come up with new triples and new non-ACT combinations. And then in the longer term, we actually have a range of options that we'd like to bring forward as potential single-dose cure combinations.
Jeremy, maybe just mention something about the single-dose cure. Why is that so important, right? And it does create tremendous hurdle for people working in the space to get the PK right, to get other property right, so that you can achieve that 7 or 8 day human half-life and for the single dose, right? Cure.
So indeed, Gang, as you say, it's a demanding target product profile, but it's one that, in the context of compliance, is really the sort of transformational agent needed to combat malaria.
And also, it's fundamental to addressing the drug resistance. If you can eradicate the parasite from the system upon infection, you would alleviate the pressure for resistance to emerge, right?
Yeah.
Yep. Great. So I'll turn the podium to Brice. Brice, you're recently moved up in MMV and took on quite a bit of the additional challenge for looking at the pipelines. So what can you tell us about some of the big bets MMV is making and the focus for the next 5 to 10 years?
Sure, thanks, Gang. And it's a pleasure to be here and to talk about MMV in front of such audience. So yeah, I'm going to touch on our, I would say, early stage discovery pipeline, and also how MMV, together with our partner, we're developing innovative approaches, or as we just call them, big bets. For example, one of them being the once monthly molecule with long half-lives for chemoprevention. We're talking about oral molecule here. So as you also discussed earlier, those molecules are raising the bar in terms of the profile we're looking for, because they need to be a very long half-life. The goal here is really to complement the current chemoprevention treatment, as well as the vaccine. As Jeremy said, there are 2 vaccines that have been approved in order to protect the most vulnerable population, such as pregnant women or women of childbearing potential. Ideally, those new drugs should have a low risk of resistance. We touch on that, and I think this is going to be a recurrent topic here for malaria. And we also want to potentially have a transmission blocking to block the passage of the parasite to other population. But one of the key things that we are exploring in this area of long-acting chemoprevention is what we call long-acting injectable. This is a whole new approach for us. And I have to say, it's also following successful models such as HIV pre-exposure prophylaxis. I'm sure you've all seen what's that in the news, the recent registration of lenacapavir, which is paving the way for us in terms of developing drugs that are long-acting injectable and staying in the human body for a while. We are very excited about that. And that kind of gives us a target that we know that it's achievable developing such drugs. However, working in this area requires quite extensive planning, including also tactical approaches for discovery and phase 1 trials, because those molecules are very long-lasting resident time. And then that's gives a lot of constraint in terms of developing the next clinical studies. In terms of other big bets, we of course continue working on the next generation of elimination tool. And here we're talking about the single dose cure for elimination. Clearly, the goal here is to have a molecule or combo of molecule that you can administer with only one tablet. And those treatment will have, of course, long duration of action, a potential low risk for resistance, because again, we just don't want to have resistance developing quickly to those new treatment. And ideally, we also want a treatment that includes some radical cure. So basically taking care of the hematozoites that are forming in the liver when you get infected with P. vivax. So that's clearly where we're going. Clearly, the bar is getting higher and higher in terms of the profile of the molecule we're looking for. But we think it's feasible, and we already have in our pipeline molecules that are showing promise in terms of reaching those target product profile. So I think
and
the next slide.
Raising the bar is a testament of how much progress we have been making in developing candidate, right? So that's the good news. The not so good news can be discouraging for new entrant to enter the field when they look at the TPP requirement. And so, I mean, this webinar is a great opportunity to let people know there's still room for improvement. Lenacapavir show you can get 6 months coverage, 99% of protection. And that's what we need in malaria, right? I mean, this.
Yeah. And we currently have early stage program that are showing promise to fit those target product profile with very long half-life. I mean, this is also depicted on this slide. I'm not going to talk again about the first generation of non-artemisinin treatment or the first generation single dose cure, because Jeremy touched on that. But clearly, for example, we have right now the first long-acting injectable for chemoprevention in malaria that is reaching the clinic, and it's currently entering into phase 1. So we're hoping that this new combo with MMV371 and MMV055, we will continue and reach phase 2 in the next couple of years and demonstrate proof of concept in malaria patients. On the side, I just want to touch base on with just in 1 minute. We also looking for other treatment, in particular for severe malaria. We have MMV183 right now that is reaching phase 1, and the goal here is to develop a new drug that could replace at one point the injectable artemisinates, and therefore be a backup plan in case artemisinin resistance developed in Africa. Then we could have a new treatment for severe malaria. And one thing that we are also quite excited about, and that is quite innovative, is that we are also looking at endectocide for elimination campaign. And in this case, we're looking at lotilaner. Here, the goal is to use the human body as a carrier of the drug. So then when the mosquito bites and take his blood meal, then he will pick up the drug, and then we will kill the mosquito via this pathway, basically. So a lot of exciting things. And again, just to show that, yes, the bar is higher, but at least we can reach the bar, and we don't want to discourage everyone. It is feasible, and I'm sure we will do it.
Yeah. The insecticide is an interesting twist in mass drug administration, right? You are giving an agent to the general population, which healthy, and it's not treating the general population, but treating the mosquito via the host. Yeah.
And I'm sure this is going to give headaches to our development team on how we're going to maneuver through the clinical phases to demonstrate the proof of concept of such molecule and how we will register it. So, but yes, it's a pretty exciting one.
Yeah, interesting. Okay. So, Elizabeth, you are one of the inventors of ganaplacide, which is, as mentioned, the first non-artemisinin based NCE in late-stage clinical development over the last 25 years. And you're also interested in, more recently, in other modalities than small molecules. So what are some of the biological target you're most excited about? And the drug resistance came up a number of times. So how can we get ahead of the drug resistant issue, which inevitably will come up because evolution is how the world evolves.
Right. Thanks. Yeah, I have a lot to say here. So as many people on this call and everyone else knows, that much of the progress over the last 20 years has been in the advancement of phenotypic screening hits, such as ganaplacide. And in fact, if you take most of our currently used antimalarials, almost all of them are derived ultimately from natural products that had so-called phenotypic activity and that they made you feel better. So on the other hand, the future is clearly with structure-guided drug design, and we can see that with lenacapavir, where they've had, a lot of detailed, information about how that was binding and how it was working. And it's just way more efficient to do structure-guided drug discovery, to know what your target is and to know exactly how your molecule is binding to its target. So in this respect, the community and also the organization which I represent, which is the Malaria Drug Accelerator, has been really focused on discovering new targets that can be used for anti-malarial drugs and in structure-guided drug design. So the way that we come about our targets is that we take a compound with anti-malarial activity, such as cladosporin or halofuginone, and then we evolve parasites until they become resistant, and then we look at the genome, and this analysis of the genome often allows us to identify the target of the compound. And this technology works really, really well, but the problem, of course, is that if you use evolution to understand the target, you've already created a resistance risk. So the case I'd like to talk about on the left is a molecule prolyl tRNA synthetase. This is a well-validated target that was discovered by my colleague, Diane Wirth at the Harvard Chan School of Public Health. And the way that they discovered this many years ago was using a natural product called halofuginone, and they evolved to resistance, and they found, that it was working through prolyl tRNA synthetase. But it was very easy to get resistance, which sort of made people wonder whether prolyl tRNA synthetase really was a good target. So we continued to work on this, and Diane has worked on it. And what we discovered is that it's only with inhibitors that bind to the proline site in tRNA in prolyl tRNA synthetase that give you the resistance problem. And that's because there's an unrelated transport, or if you knock it out, you suddenly increase levels of proline in the cell, and this leads to resistance. However, if you're targeting the non-proline site, like the ATP binding site, you don't have this resistance problem. So I really love this because it just shows how, developing a detailed understanding of how compounds are working and how they're binding can potentially lead us to, better inhibitors that don't have the same resistance risk, and that this problem about resistance isn't necessarily with the target, but it's often, with the particular compound and its binding mode. And it also shows, that there's still a lot that we need to do in terms of basic biology, and sometimes serendipity can lead to really important breakthroughs in drug discovery. I'd also like to mention that we now have a much better understanding of what leads to drug resistance than we had a few years ago. It used to be kind of a black box thing, and it's just, oh, this drug's not working. And one of the things we've been doing through the Malaria Drug Accelerator is compiling all of the different ways that parasites can become resistant to small molecules. And so these are often mutations in transporters, but they're also mutations in targets. And we can start putting them together and begin to really develop an understanding of how the parasite is becoming resistant. And, one of the studies we published last year on, where we talked about the resistome, we started looking at the features of resistance conferring mutations and showed, for example, that they're often in particular domains that interact with inhibitor. There are specific types of amino acid transitions, so from a small residue to a big bulky residue, for example. And we can start putting this together to start, hopefully developing artificial intelligence and machine learning approaches that could potentially augment, compound design. So I think the future, right now we're using artificial intelligence to design compounds that can bind, but I think, maybe 5 years from now, we'll be able to use artificial intelligence to predict all the different ways that a target can become resistant and to start incorporating that into structure-guided drug design. So yeah, that's where I think we're going. But of course, it probably take a number of years before, these current, these targets, ultimately lead to new drugs. But, there's a lot of room for doing good biology at this point.
Yeah, Elizabeth, the pro tRNA synthetase is a great example. I'm somewhat a converter now because previously I had this narrow mind of if a molecule shows very low MAR, that's probably suggests the target's not good. But now we have more examples that I can count on my finger that you can definitely improve the MAR from 10 to the 6th all the way to greater than 10 to the 9th, which is considered to be refractory to resistance. And it does require a tremendous amount of structural biology information to guide the design. And with AlphaFold3 and cryo-EM, that's getting easier to do. Great. Maybe we can switch gear to talk about some other modalities than small molecules. Small molecules certainly occupies a big part in my heart, but the drug universe is much bigger. And as the vaccines have been mentioned, but there are also other activities. Brice, you've been kind of wading into that space, and Elizabeth, you too. And Brice, you start.
Yeah, sure. As you say, there currently are 2 vaccines out there registered. So of course, we've not been part of this development as MMV. But our strategy now is that we need to consider the vaccines and their impact. And then it shows also that biologics overall could have a utility in malaria, of course. And in the recent year, I mean, MMV has been looking at what's out there beyond small molecule that could be of interest to help us in the malaria field. So at MMV, we've kind of constituted a small team, and I'm now looking at all the technology that we could investigate and see if we could demonstrate any proof of concept that they could really be helpful in malaria and help us in the chemo prevention world. Why are we doing that? Is mainly because, okay, we're trying to get the best of the two worlds, the best of the vaccine world together with the best of the small molecule world. We know that each area have the pros and cons. In the small molecule world, they are very interesting because they are affordable, they are cheap to make, very highly efficacious, but of course they can raise resistance. In the vaccine world, you also have some good efficacy there, but mainly their main advantage is that they can be administered to the population in need, in particular in pregnant women and women of childbearing potential. But in the vaccine and now also in the monoclonal antibody wars, which are the kind of the next generation biologics, the problem is that they are not cheap to make. They are pretty expensive, and logistically, they need also some cold chain. So our thinking was, what can we find out there in terms of technology that could help us gather the pros of each world? And in this way, in this regard, what we found is that we now currently investigating a few projects which are looking at nanobodies. We're also looking at new monoclonal antibodies, and in this area, what we're looking at really is using self-amplifying DNA or RNA expressing monoclonal antibodies, for example. So we just recently signed a contract with a company, and we're going to explore that a little bit further. We're also looking at antisense oligonucleotides, and I'm sure Elizabeth will talk about that. And we're also looking at new small protein scaffold, which could also be of high interest for malaria. So the goal here again is trying to find biologics that could be cheap to make, and therefore the cost of goods could be low. The biology that could be highly potent and effective, that would not need the logistical chain, such as needing a cold chain. And right now we're just at the beginning, but we're already building a small portfolio of projects there that we're planning to at least investigate in the next few years.
Yeah, thank you, Brice. Even in the context of having highly efficacious biologics available, I think there is still this scenario that you want combined with small molecule in the beginning,
yeah,
clear the parasite in the asymptomatic population, so you will have a clean background to start for the chemo prevention.
Yeah. And you're right to mention that, Gang, because again, what we're saying here is that there's no one way or the other, is that every single intervention will be needed together to combat malaria and reach elimination. It's not only one intervention that will do it.
Yeah. So Elizabeth, antisense oligonucleotides have been proved, marketed for a number of disease indications. Nothing has been used for infectious disease. So tell us about why you're interested in ASOs and how is the project going?
Well, yeah, so antisense oligonucleotides, for people who aren't familiar with them, they're nucleic acid technologies in which the backbone has been modified. And there's a lot of different modifications that can be made. So the molecule will still bind to its target, which could be possibly genomic DNA, or it could be a transcript, and it would be very sequence specific, but it would resist degradation in the human body. And so this, as we start really thinking, long term about things where you'd have a single injection and it would protect you for 6 months, antisense are quite attractive in that respect, because they're naturally long acting. For example, Spinraza, which was just licensed for spinal muscular atrophy, that's treated with a single injection every 4 months. And so, and the cost, it might be a problem, but the ASOs are typically stable at room temperature. We have a variety in the lab, and they just sit out in the fridge, and you don't really have to worry about them, getting degraded in ways that you would if you were dealing with RNA or DNA. So that's super attractive. However, I think there hasn't been a lot of work done on antisense technologies at this point, and there's a lot of opportunity for exploration. There have been a few studies published, but I think a lot of stuff is unclear. So hopefully you'll see a lot of work in this area or coming along, and maybe the next long-acting injectable that will be functional for chemoprevention may be an antisense technology. I'd like to add one more thing, though, about the monoclonals, though, before we move on, just because, it seems that they often don't work quite as well when you go into field situations. And I think it's going to be really important to understand resistance risk in the organismal context, just like, we're studying this in with small molecules, if we really want to make monoclonals that work, for the long term.
Yeah. Another interesting fact about ASO is they selectively accumulate in liver, and liver is the beginning of the parasite life cycle.
Right.
And I think that make it a very natural fit for the intervention using ASOs.
Yeah.
Excellent. So, I mean, there are so many innovations we can look forward to in terms of delivering the tools for elimination and eradication hopefully in 15, 20 years. Let's switch gear and talk about roles of open science, open data, and of course, artificial intelligence. People recognize that there's limited resource in global health R&D, and we really have to do things smarter and more efficient. So, Dom, what is MMV doing in these areas with regard to open science and open data?
Thanks, Kang. Well, MV is doing a lot. And MMV started or embraced the open research strategy since quite a long time now. It started in 2012 already, when MMV has created its first of the 5 open access compound boxes to help scientists accessing freely reference molecular compounds to initiate research program. And since that time, plates of those 5 boxes were shipped to scientists in more than 50 countries around the world, which I think underlines the importance of such an activity. Gang, may I ask you just to click once? Thank you. So, so MMV is it is not the only initiative that MMV has taken. So the first one is, together with our donors, we have built compound libraries to identify novel starting point for medicinal chemistry projects. And we have acquired those libraries, but we have openly shared these libraries with the other PDPs and partners, so that it's not only focusing on malaria, trying to open the research to other pathogens. In parallel, MMV is now proposing several open research initiatives, such as the Collaborative Drug Discovery Program called Malaria Libre, or the Malaria in Vivo testing setup called MMVOI. On the technology side, we have developed and are still developing software that must help, first of all, the malaria community to access freely prediction interfaces. We have 2 existing examples that are, on one side, MMV Solar that is proposing free access to dose-to-man prediction functionalities, and the other one is MMV Free, an interface for malaria chemoprevention, helping users to predict molecules and bond properties. Now, this is great, but if we want to look a bit wider and to access the larger community, we need to slightly increase our activities there. So today, MMV is active on several open research projects. And from these projects, I will mention two that are very close to my heart, I would say. The first is a kind of mentorship program supporting disease endemic institutions, and more specifically, African scientists, in order to develop their own open access compound box, but a box that is focusing on African natural products. Another one on the AI ML front, it is very important, if not critical, to mention the DD4GH project. This project aims to grant global health scientists a free access to prediction models and machine learning capabilities. Why? Because at the MMV, we believe that AI and ML will be fast-track research program by improving early drug design and molecular properties of produced compounds. So today, MMV is not the only institution proposing open access programs. And I know, for example, that Mariana, CDD Vault is as well active in opening access to its database to African scientists. Am I correct?
Yes, of course.
Yeah, Mariana. Yeah, you guys are super passionate about enabling open access science. Yeah, talk about your activities.
Yes, very passionate about that. Thank you, Dominic, and thank you, Gang. Yes, I agree with you. Open science is key to drug discovery, especially in tropical diseases, right? Where knowledge should be shared and data should flow as freely as possible, if you want, across borders, but also across disciplines. And collaboration with a bit of competition should be key to solving some of these key global challenges. I think this is, in this kind of collaboration is where CTT mission aligns very well behind all these initiatives. And our main initiative is the African program. And this is kind of main objective of this initiative is to level up the technology playing field. Too often, access to the latest technologies is confined to the well-funded institutions in well-funded countries. And CTT aims to ensure that scientists across Africa have the same advanced tools to manage, to analyze, and to share the research. In that context, we have this grant program. So I'm going to pass my little ad here. So if any African scientists are listening, please contact us. It's a grant program that that will enable you to access our CTT Vault. If you are working on malaria or tropical diseases, you will have access to the technology and all our support. Simply reach to us and we will make sure that you are organized. Since the inception of collaborative drug discovery by Dr. Barry Bunin, our CEO, CTT Vault was envisioned like not just a software tool, but almost as a mission-driven platform. And the mission is to be a catalyst for collaboration and a bridge between disciplines. And so please join the program if you can. And yeah, we enjoy it. We're very passionate about this. So, yeah.
Thank you, Mariana. Thank you, Barry, for providing the in-kind support to our African researchers, which is making tremendous impact. So let's spend some time on artificial intelligence. We're 2.5 years post the initial ChatGPT 3 and 3.5. And it's probably good to say that AI has been making impact on drug discovery and development, although the impact has been uneven, quite uneven to say the least. Kelly, can you highlight the pharmacogenomic work you're really passionate about and working with the pharma partners?
Yeah, thank you, Gang. Yeah, I think this is something that is very close to my heart. When Mariana was talking about just the impact of AI, and of course, it's really this technologies I think is democratizing science and helping people to leapfrog progress. So Jeremy, after me, will really unpack how AI is really making a difference in the malaria drug discovery. But let me focus on just the aspect of pharmacogenomics. In other words, what is the impact of genetics on response to medicines? That's the overarching goal of this project we've done under a program called Project Africa Gradient. And this is a program that was started by GSK and Novartis. And we participated in this program on a project that was using AI to help us leapfrog the barrier of not having, for example, biobanks in Africa, but use a technique we call transfer learning, especially from the oncology field. But use AI to identify genetic variants which are prevalent in Africa, which are likely to affect the metabolism of malaria and tuberculosis drugs. This was the study was focused on a pilot project looking at malaria and TB drugs. And then once those genetic variants are identified, they are then incorporated into existing mathematical models that we use in predicting the human dose. So an example is the physiologically based pharmacokinetic modeling, PBPK. And so incorporating the effects of these genetic variants then allows us to propose tailored dosages of these malaria drugs, for example, in people carrying these particular genetic mutations. So this is something we've completed this project last year with GSK and Novartis, but also with the South African Medical Research Council. And we have a paper under review, which was deposited into this link that you can see on this slide. But I'll turn over to Jeremy that he can really unpack how AI is being used in malaria drug discovery for the benefit of the audience. Jeremy, over to you.
Okay, thank you, Kelly. So yeah, let me touch on some of the areas in which AI can have an impact, really. So it begins with chemists being able to bring their structures and their data into tools where that data can be modeled. And then the next sort of really important, exciting step is the generative methods to actually create virtual structures which then have their properties predicted against the said models. Then we can actually take these predicted these virtual structures and triage them based on these predictions and all these properties. We can indeed select actually those which we can make in a reasonable time frame, because we want this make test cycle to be weeks, not years. And we can even test for relevance. And from our perspective, we passionately believe in optimizing on dose. And as Dom had mentioned, we've delivered various tools like MMV Solar, MMV Free, which effectively help us to do that. And then there's the analysis stage. And this, in fact, can sort of come automatically with updated auto modeling. And then the idea is that these compounds which have been selected to be made and tested. Once that data is in and fed back into the models, and the models themselves improve. So you not only end up with better compounds, but better models for that next design cycle. And I guess really, you know, predicting properties, we believe will improve quality. And by selecting compounds that really teach us, you know, something new, we will make every compound count. I'd love to be able to say that, you know, that with all of this strategy, we can, you know, deliver a drug in you know, in 6 months or something, but that's not the case. That's not the case yet. And at the moment, we're really looking to bring this into practice to, for instance, save a year between delivering a late lead from a hit.
Yeah, so I think talk about, Jeremy, some of the initiatives that will enable the faster DMTA cycle and deliver the therapeutic candidate faster and the higher quality.
Okay, so Dom had mentioned the malaria inhibition prediction tool, and this is a open access tool on Campo where you can enter a virtual structure and you get a number which basically indicates the likelihood of it being active on malaria. But the key piece for us is this tool, DD4GH, which stands for Drug Design for Global Health. We're working with a London based AI company called Deep Mirror, of a very nice collaboration. And basically this Deep Mirror DD4GH tool is targeted to deliver exactly what I just actually went through in terms of modeling the data, virtual chemical design, triaging, active learning. And we're in the process of sort of tuning this and, you know, improving and increasing features. We will have DD4GH delivered by the end of March next year. As Dom said, this is going to be freely available to scientists working in global health. And, you know, just to emphasize Kelly's point, it's so important for us to democratize machine learning for Africa, really.
Yeah, it's also worth mentioning the DD4GH is disease agnostic, right? Even though MMA is building this in collaboration with Deep Mirror, it's not limited to malaria. It should be applicable to TB, to NTD, to other, right? Yeah.
To any pathogen, absolutely. Thank you.
And Mariana, maybe a minute to highlight the CDD Vault capability.
Yeah, and we definitely believe that AI, of course, is a systemic change in research. And we incorporated a lot of AI tools into the platform, from idea generator to predictive folding and ligand docking tools. But I agree with Jeremy. What makes the technology truly transformative is that we can make it accessible to scientists, so easy to use and accessible, so they can test their ideas even before they go into any lab. And it's, you know, it's a radical change in the way that we do science right now. And CDD Vault is in the middle of that and fully implemented, so people can use it today. So scientists can start using it today. So yeah, looking forward to see the results, and maybe 6 months pipeline delivery.
Great.
I have to.
Thank you, Mariana. Maybe I'll turn the table over to Kelly and James to talk about doing drug discovery in Africa, because previously it was mentioned 94% of the malaria cases happen in Africa, but most of the R&D work are being done in global north, which is not a terribly good picture. So Kelly, how can we build that capacity on the continent in Africa? How do we enhance the community engagement?
Yeah, thank you, Gang. So I think from my perspective, I think there are three main reasons why it's important to do R&D in Africa, broadly speaking. And of course, importantly for malaria, for the reason that you just mentioned, we bear the greatest burden of this disease. So the first reason is really that, as Africans, we have to contribute solutions to this problem because, of course, we are the most affected. But of course, it's not a solution that we can come up with on our own. This has been reflected in this very webinar where we are working together with various partners. So firstly is for Africans to participate in R&D, to contribute solutions to this very significant unmet medical need. The second reason is that there is a very direct link between the genetics of a population, the social and physical environment in which those patients live, and treatment of disease. And this is very important because, and I'll come back to this a little bit later, when it comes to genetics, the African continent is the most diverse genetically. So it really makes a lot of sense that to do the discovery part of the process and the development part of the process in close proximity to where the patients are in their social and physical environment is the best way to address these unmet medical needs. The final reason for why it's important to do R&D in Africa is exactly the one that I mentioned of this variable genetics that we see. One of the reasons for the variability in the way people respond to medicines is a consequence of genetic differences in the expression and activity of drug metabolizing enzymes. This has not been well understood in the African context for two reasons. One, we have a very low volume of clinical trials that happen on the continent, roughly about 4%. And yet, malaria, as you said, Gang, 94% of the cases are on this continent. But the other reason for this is that in the preclinical discovery phase of the program, there are no tools available. For example, when we buy microsomes or hepatocytes in drug metabolism, those come from Caucasian donors, not from Africa, which means that we don't have a representation of these African livers, which are so diverse, will allow us to prioritize small molecules based upon their predicted profile in African patients. And then finally, before James talks about the same issue, community engagement. Any kind of innovation without access is pointless. When it comes to drug discovery and development, we have to bring communities early on in the process. For example, in the discovery, they need to know, they need to have input into what is coming their way. For example, Brice was talking about long-acting injectables, and I'm sure MMV did this to understand whether this will be acceptable to the communities that will take these tools. So we have to involve them right from the beginning in the discovery phase so they know what's coming. And also we build the trust that we need to build. James, I'm sure, has words of wisdom to add to that as well. James, over to you.
I have words to add, Kelly. So yeah, thanks for summing up the situation so eloquently as ever. I mean, for those of you looking at the slides, this is just this is a snapshot of some of the partners that MMV are working with on the African continent at the moment. The list is always changing. To be fair, it's always growing, and this is great because we're absolutely passionate about R&D in Africa being a pillar of our approach towards malaria drug discovery. It's actually, it's a solution, not just a problem. And what we really feel, and I was able to observe this, I was fortunate enough to spend a year on secondment as part of my role in MMV with Kelly and his team. And we could see, and I could see firsthand the absolute potential of African scientists, their unique motivation, their perspective, their energy, and their passion to discover new drugs that disproportionately affect the continent is second to none. And once you harness that, you realize that you can leverage the knowledge, the skills, the experience, in so many cases unique to these scientists, especially in terms of some of the assays that they can perform, that can only be performed effectively on the continent. And once you appreciate this and bring this into your thinking and bring this into your process, you actually start building a virtuous circle where you create success, and that success creates more funding, creates more interest, creates more opportunities, which builds more success. And this goes on. We realize that we're at the beginning of these cycles, but every time we go around this cycle, we see another significant impact of this discovery, and this can only grow and will only grow. And I think with leaders like Kelly sort of pushing this forward and this agenda forward, you know, it's an unstoppable force. So that's why we're fully behind this.
Thank you, James. Thank you, Kelly. So if we take a step backward and look at the global picture, right? Elizabeth, you and I build and are co-leading the Malaria Drug Accelerator. It's a collaboration on the global scale, including African researchers. Maybe a couple minutes to just introduce and share what Malta is doing.
Yeah. So Malta is involved in the very early stages of drug discovery, including target discovery, and we also evaluate early stage phenotypic screening hits as well. And this is a consortium of, was it 19 labs at this point? Sorry, I'm losing track. But we're spread all over the world, and we operate on the principle that resources for malaria drug discovery are very limited. And therefore, it's really imperative that we have this organization where we can share what we're doing, share the identity of molecules that we're working on, share the identity of targets that we're working on, so there's no duplication of effort. I know if you're in a pharmaceutical company and you're working in a really competitive area, maybe you say, well, we don't want anyone to know what we're doing, because if other people know what we're doing, that increases competition. But, we're not in this situation with malaria. I mean, it's just such a small pot of money that's available. So we have to talk to each other. We have to, optimize our resources. We have to, to use things as efficiently as possible. We can't silo it. We can't, hoard resources. And so Malta is an organization that really helps us to do this. And, we meet and we talk to each other. And we also try to share everything that we're doing with the rest of the community as well. And so, yeah, that's where we're going with Malta.
Yep, I mean, sharing and collaboration are the key, and we have agreement in place to govern the processes.
Yep.
And it's working very well. Okay, we're getting to the end of the webinar. I would just ask some of our speakers to leave a departing wisdom with us. And where do you see malaria drug discovery development in 5 to 10 years? If you can, Kelly, Elizabeth, and then Brice. Keep it short.
One quick one from me. Many things I'd love to say, but one area for me will be polypharmacology. I think we had right from the beginning, from you, Gang, from Elizabeth, from Jeremy, resistance is always going to be the biggest need. So I think for me, it's going for small molecules, single agents that can affect the parasite at multiple stages is the way that I see the future of malaria.
Great. Elizabeth?
Well, I would say my future is more, I put up this picture of lenacapavir, bound to the HIV capsid, because, I think the future is also, having enough knowledge about your target that you can build these super selective inhibitors and at the same time mitigate resistance risk, while at the same time making something that is, can be in the body, that protects you for 6 months. So that's where I think the future is going.
Brice?
Yeah, I won't reiterate the thing about resistance. For me, that's also a key thing for the future, but I would also highlight the long-acting injectable area. Again, lenacapavir has paved the way. So it shows that it is feasible. It's a high bar and a high target, but we can do it. And I think this would be a massive improvement for malaria. And of course, maybe biologics also could be a very helpful tool in the future, in particular in terms of safety for, again, the population in need, such as pregnant women or women of childbearing potential.
Excellent. We are right on time, and we have the direction of travel. So much work to do, and I look forward to collaborating with every one of you on the ongoing basis. So Joshua, over to you to close out.
Thank you so much, gang. We appreciate it. And thank you all to all our speakers and all our fantastic panelists for that discussion today. We will be sharing the slides and the recording in a follow-up email, as well as getting to the Q&A questions that we were not able to get to, didn't have time for today. So we will please look out in your inbox for that email. And thank you to you, our audience, for attending today. Hope you enjoyed the conversation, and thanks again to our speakers. Have a great day, everyone, and have a great rest of your week. Cheers.
Thank you.
Thank you.
Bye now.
Bye-bye.
Everyone. Thank you.